Hey all, here is the chapter 20 overview about phylogeny. So phylogeny is the evolutionary history of a species or group of related species, and we use systematics, which classifies organisms and determines their evolutionary relationships. So within that, we use taxonomy, that's the ordered division and naming of organisms. Linnaeus is the scientist who came up with that system of taxonomy, and it's the two-part names for species. So we still use that today with the scientific names of organisms. So it's called a binomial. The first part of the name is the genus, and then the second part is the species. And just when writing it, the first letter of the genus is capitalized and the entire species name is italicized. And both parts together name the species, not just the second part, not just the species alone. So the taxonomic groups from broad to narrowest, meaning only one individual species has that name, is domain, is the first overarching group. Then there's kingdom, phylum, class, order, family, genus, and species. So a way to remember this is dear King Philip came over for good soup. Maybe King Philip came over for something else that starts with an S. Just however you remember it. So dear King Philip came over for a good soup. All right, so that is the order from broadest category to most specific. So here's an example. Down here, this is broad going up to specific. So down here would be the domain of Eukarya, and then kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Felidae, genus Panthera, and then this is Panthera pardus. So one species here, there would be all eukaryotic organisms would be in Eukarya, and then kingdom Animalia, all animals. So as you go up in this instance, it gets more narrow until we're talking about one individual organism. So a taxonomic unit at any level is called a taxon, and then the broader taxa are not comparable between lineages. So, for example, an order of snails has less genetic diversity than an order of mammals. So you can't really compare between the two. All right, and then we can depict evolutionary relationships in phylogenetic trees. And we can use those and look at where different either characteristics came to be, shared derived characteristics, so they arose during time, or shared ancestral characteristics. We can also look for common ancestors. So right here, anywhere where there's a branch point, that means there was a common ancestor here that branched off into two other species. Back here, this would be the common ancestor for all of the species on this tree. The closer two species are together, the more closely related they are. And we can turn this. So this can be on the side like this. We could flip it so that this branch at the was at the bottom instead of on the side. We can do it on an angle. They all mean the same thing. So a phylogenetic tree represents a hypothesis about evolutionary relationships, and each branch point represents the divergence of two taxa from a common ancestor. So two species from a common ancestor. And then sister taxa are groups that share an immediate common ancestor. So these two are sister taxa, these two are sister taxa. A rooted tree includes a branch to represent the most recent common ancestor of all organisms in the tree. And then a basal taxon diverges early in the history of a group and originates near the common ancestor. So this right here, this would be our basal taxon. A lot of times we call this the outgroup. So this is the group that arises early on, and a lot of times we use this in comparison. So again, this branch point is where lineages diverge. And then here we have sister taxa, and then again, this is our basal taxon. So phylogenetic trees show patterns of descent, not necessarily phenotypic similarity. They don't generally indicate when a species evolved or how much change occurred. And it should not be assumed that a taxon evolved from the taxon next to it, right? They share a common ancestor. They typically arose independently. Phenotypic and genotypic similarities due to shared ancestry, remember, are called homologies. So we can use those homologous structures to put organisms on a phylogenetic tree. So organisms with similar morphologies, that's bodies, so those homologous structures or DNA sequences are likely to be more closely related than organisms with different structures or DNA sequences. When constructing a phylogeny, systematics needs to distinguish whether a similarity is the result of homology or analogy. So remember, homology or homologous structures are similarities due to shared ancestry, but remember, analogies or analogous structure is similar due to convergent evolution, and these analogous structures do not indicate that those organisms are closely related. So convergent evolution occurs when similar environmental pressures and natural selection produce similar, which are analogous adaptations due to the environment. So these two organisms both are underground, right? They both burrow. So you can see they're shared, they have these claws here that are useful for digging. Their noses are big, but they are not closely related. They just share similar structures due to the environment that they live in. So bat and bird wings, again, are homologous, but homologous as forelimbs, so they share the same structure. We can compare fossil evidence and the degree of complexity to distinguish between homology and analogy. So the more complex two similar structures are, the more likely it is that they're homologous. And once homologous characteristics have been identified, so once we know that they're homologous, then they can be used to construct a phylogenetic tree. Cladistics classifies organisms by common descent, and a clade is a group of species that includes an ancestral species and all its descendants. So clades can be nestled or nested in larger clades, but not all groupings qualify as clades. So a valid clade is monophyletic. Mono is one, signifying that it consists of the ancestor species and all its descendants. So typically when you look at phylogenetic trees or cladograms, they're both very similar. You're going to see this common ancestor here. So as long as we see this common ancestor for everything on the tree, this is a monophyletic group or a clade. And we can even divide that up into, if we're looking just at this group one here, this is also monophyletic because this number one, that's the common ancestor for A, B, and C. There is also the possibility of you looking at different groups. So if we're looking at group two here, this is a paraphyletic group, which means that we have this ancestral species 2 right here. We have some, but not all of its descendants. It's leaving G out if we're just talking about group 2, so that's called paraphyletic. And then polyphyletic is a group consisting of various taxa with different ancestors. So because in group 3 we included D here, we have 2 common ancestors, more recent common ancestors. So in comparison with its ancestor, an organism has both shared and derived characteristics. A shared ancestral character is a character that originated in an ancestor of the taxon, meaning everything in the tree that you're looking at has that characteristic. A shared derived characteristic or character is an evolutionary novelty unique to a particular clade. So when we're looking at the tree, we can look at or figure out how to construct a tree based on derived characteristics. So when we're looking here at the character, we can see backbone, hinge jaw, forelimbs, amnion, which is like amniotic fluid. This could either be in a placental animal, or amnion is also in eggs, milk, and then a dorsal back fin. So when we're looking at the chart, we look to see which characteristic probably arose first, which means most of the organisms will have it. So if we look, backbone, all of these organisms have a backbone, except for the lancelet. This is the outgroup. So we would know a backbone would arise first, and then hinge jaw, then forelimbs, then amnion, then milk, and then a dorsal back fin. So we can use this to construct. This is another way you may see a character table. So we could have ones here, meaning they have it. We could have pluses and minuses, same thing. This is just organized a little bit differently so that we can see where all these characters arose. This one has hair instead. We can use that then to create, and you guys will need to create a phylogenetic tree. So you would put those derived characteristics on each branch point where they arose. So the lancelets are the outgroup. They don't have any of the derived characteristics. Then the lamprey comes next because all it had was the vertebral column. So that goes here. So then everything else on this phylogenetic tree is going to have vertebrae. Hinged jaws is next, meaning everything but the lancelet and the lamprey have hinged jaws, then 4 walking legs, then amnion, and then hair. So in this way, by looking at these derived characteristics, then you can determine where all these organisms are placed on the phylogenetic tree. Again, the outgroup is the group that's diverged before the in-group. The in-group is all the species being studied. So typically the outgroup is not going to have the derived characteristics of the rest of the clade. And we can compare each in-group with the outgroup to differentiate between shared derived and shared ancestral characteristics. All right. So in some trees, the length of a branch can reflect the number of genetic changes that have taken place in a particular DNA sequence in that lineage. So we can see in this picture here, this shows the length of time. So you can see how some of these lines are really long. So that can just indicate the number of genetic changes that have taken place. Because this one is really long, this means there was no change, right? It just branched off really early on. This is just showing that everything is alive now, is why that branch length was so long. In other trees, branch length can represent chronological time, and branching points can be determined from the fossil record. So typically the difference between a phylogenetic tree and a cladogram, phylogenetic tree shows the amount of change over time. So if we're looking at a phylogenetic tree, a lot of times this will show time, where a cladogram just shows the relationships between organisms. So again, we can see that human and mouse are the most recent in history. We're Drosophila, we're way back. All right. The other thing we need to talk about is maximum parsimony. I know this is a weird kind of vocabulary word or words. So that just assumes the tree that requires the fewest evolutionary events is the most likely, right? So the simplest explanation is typically the correct explanation, and that goes for almost everything in the world. So what we look at, so let's say we have these 3 species of birds. We don't know which one arose first. We're not sure where to place them on the tree. So we look at all of their shared characteristics, or here we're comparing sequences of DNA. And when we look, we see the results here. So we had 3 possible hypotheses. And when we analyzed the DNA, we realized that this first tree only took 6 events compared to 7 events of the second and third hypotheses. So we go with this first hypothesis. The least events possible, the simplest explanation is how we construct the phylogenetic tree, which means typically we go with the derived characteristics in order, so that something isn't losing something, gaining something, losing something, gaining something. It's just things don't have whatever trait it is until it arose, and then everything beyond that point has it. There might be a point where that trait is lost in a branch, but it is the simplest explanation possible. So the best hypotheses for phylogenetic trees fit the most data. So we want to look at the most data possible: morphological, which is again structures like homologous structures; molecular, meaning DNA or proteins or amino acids; and then fossil records. These can be modified. So we come up with these phylogenetic trees. They are hypotheses. They can be modified if we find a new fossil, if we're able to recover DNA from an ancient species. And bracketing allows us to predict features of ancestors and their extinct descendants based on the features of closely related living descendants. So an example is we can infer characteristics of dinosaurs based on shared characters in modern birds and crocodiles. So here's a common ancestor of crocodiles, dinosaurs, and birds. We have this phylogenetic tree. So we may be able to come up with what we think this common ancestor, what characters they would have. So birds and crocodiles share several features like a four-chambered heart. They sing, they have songs, nest building, and brooding. And these characteristics likely evolved in a common ancestor because they share all of those traits. And the fossil record supports nest building and brooding in dinosaurs. To extend molecular phylogenies beyond the fossil record, we have to make assumptions about how molecular change occurs over time. And we do this by using a molecular clock, which uses constant rates of evolution in some genes to estimate the absolute time of evolutionary change. The number of nucleotide substitutions in related genes is assumed to be proportional to the time since they last shared a common ancestor. And molecular clocks are calibrated by plotting the number of genetic changes against the dates of branch points known from the fossil records. Individual genes vary in how clock-like they are, so we tend to use neutral mutations. So because those neutral mutations have little or no effect on fitness, they should mutate at a standard rate. So that neutral mutation rate, though, is dependent on how critical the gene's amino acid sequence is to survival. So when we look back, here the number of mutations, if it's a neutral mutation, tends to be steady, and then we can figure out the divergence time in millions of years. There are a few potential problems with molecular clocks, so they don't run as smoothly as expected if they were selectively neutral, so sometimes it creates some irregularities. And we can have some irregularities because there may have been natural selection occurring. Estimates of evolutionary divergences older than the fossil record have a high degree of uncertainty because we can't use those fossils to check to see if we're correct. A lot of times then they'll use multiple genes, and that can improve estimates. Best data to use when constructing a phylogenetic tree is similarities in DNA or amino acids. That is data that we have, it's specific. So if we have DNA or amino acid data of how similar or different DNA sequence is, we use that. Another somewhat recent development is we went from 2 kingdoms to 3 domains. So now we use domains as the broadest category. So early taxonomists classified all species as either plants or animals, but then later there were 5 kingdoms. More recently, we've adopted a 3-domain system: bacteria, archaea, and eukarya. So you can see domain eukarya is plants, algae, fungi, and animals. And then archaea is ancient bacteria. And then we have domain bacteria, which is all of the bacteria, all of the other bacteria. All right, that's it. Really, to understand phylogeny, phylogenetic trees, and cladograms, you guys have to practice putting those trees together based on derived characteristics or shared amino acid or DNA sequences, which we will be doing that in class. All right, hope that helps. Bye.